Alright, so far this unit we talked about RNA and what it's needed for, how it's made, how it's built, and then we've also talked about transcription, taking that RNA and making it from the DNA. That's the whole process of building RNA, the whole process of transcription. Today we're going to look at the next process, and that's translation, where we ended the other day. So we're going to look at translation. Translation, there are quite a few more steps here. So we have 9 steps to translation. All right. We've already had that RNA made. It's left the nucleus, and it has to go to the place where proteins are built. Where's that? That's the ribosome. All right. So the first step is that mRNA goes and it finds a ribosome. Once it finds it, it binds to the ribosome, and the ribosome uses it kind of like it's a decoder basically of that strand. That strand gets pulled through the ribosome, and the ribosome decodes it as it's pulled through. Okay, so the ribosome looks for a place to start, and the start codon always is AUG. AUG is the place that the ribosome finds. It finds that codon, those 3 bases, and it says, okay, here's where I start decoding this protein. Okay, so that's the start signal. That AUG codes for methionine. That's always our start codon. All right. The next step is the ribosome has it's going to match this up. So we have that 3 base codon AUG. It's going to find a tRNA, transfer RNA, that is the exact opposite, the complement to AUG, which would be UAC. That's going to complement it. And so it brings in that tRNA, it attaches there. Now, on that tRNA, we have an amino acid. There's an amino acid attached to the end of that tRNA. And so as it brings it in, it brings that amino acid in, it attaches, and then the ribosome moves to the next 3 bases. And it finds the tRNA that fits that. And once those 2 are locked into the ribosome, the amino acids are linked together. Okay? And we'll look at that in just a minute, how that process works. Once that first one, that methionine, is linked to the second one, that tRNA leaves. It leaves the ribosome, and the whole thing slides down. It shifts down for the next one, and then the next one, and then the next one. And it just keeps building and building and building until it reaches a 3 base sequence that says stop. Okay? So you should have printed out or downloaded the codon charts, and that's how we're going to decode this stuff as we go. All right? Once we have all of those linked, and they're linked by what we call peptide bonds, all right, peptide bonds, we have a polypeptide, many peptide bonds, bonding all of these different amino acids together in a long chain. That chain is called a protein. All right. So polypeptide right here is another name for protein. Okay. Now this process seems kind of crazy, but we're going to go through it a little bit and you'll see what it looks like. Alright, so moves down, keeps going till it reaches a stop. Now that stop codon tells it you're done, that protein's complete. It stops it, the protein gets sent to the endoplasmic reticulum. It modifies it, sends it to the Golgi apparatus. All those different things that we talked about way back when come into play here. Okay. Once the gene has been decoded, the mRNA is useless. It's trash. All right, and it goes away. So what happened here? We had our first codon got read, and it was it's not the first one, but TYR, tyrosine. Then the next one got read, asparagine. And now we're at this point reading this one. And we're inside the ribosome. The ribosome finds this tRNA, and notice it's called tRNA because it looks like a T. Okay? tRNA, it's got arginine attached to it, and it holds in this ribosome until the ribosome finds the next one that matches. Once that tRNA comes in, then this one will be released from the tRNA. This tRNA will leave, and it will bond to the next one. the next amino acid. So we have amino acid, so on, until it reaches one that codes for stop. And that means the protein is done, it is built. Okay? Now, looking at a tRNA, it's a little bit different picture of it. They don't always make them look like T's in the picture, but in real life it does make that kind of a T shape. This piece down here, we have 3 bases down here that attach to the mRNA inside the ribosome. Well, if the mRNA has 3 bases called a codon, the opposite of that is an anticodon. All right, so on the tRNA we have an anticodon. We have our structure here of the tRNA, and then we have our amino acid attached to the top. Okay, that's pretty much it for tRNA. It's just there to bring in this amino acid. That's all that we worry about. We don't care about the structure. It doesn't matter. We know that this is an anticodon that's opposite to the codon, but the most important part is that it brings in these amino acids. Okay. So you should have printed this or have it downloaded. This is one of the charts. There's a square one, there's a circle one. You've got to be able to use both of them because we have no clue which one they're going to put on the STAR test, so you got to be able to read both of them. How you read this, and we're going to go over the square one here. How you read this is pretty simple. Okay, so first you're going to be on the left-hand side over here, and it says first letter. All right, so let's start with the start codon, AUG. So if we're going to do AUG, we find the first letter over here is A. That means that we're going to be in these rows right here. Okay, we're going to be in these 4 rows. The next one is the U. So we go to the top, second letter. Here's the U. That means we are going to be in this column right here. Hard to draw on this thing. This column. So where those 2 intersect now are just these 4 right here. Isoleucine, 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 methionine. So our last letter is G. We go over here and we find G, third letter on the right side, which means we are going to be using methionine. AUG, methionine. Okay? Makes sense? If we did U, G, A. All right. Here's my U right here. I'm going to star this because that's the one we're doing now. Here's my G right here. Here's my A right here. Where do all 3 of these meet? All right. We go across. We go down. We go across. We find that UGA is a stop codon. Okay. You got to be able to use these charts. Make some up. You're going to have some practice today. Practice coming up to where you start reading these charts so that you know which amino acids are going to be in each sequence. Okay. So what happens to that mRNA when the sequence is over? All right. Once it reaches that stop codon. So we just did a stop codon. We just did UGA. So there's a stop codon. It hits that last one. The protein is built. No more need for that piece of mRNA. Okay. Stop codons don't code for anything. It's a signal to say stop. Right there. Okay. The protein gets folded up, gets sent to the ER. ER modifies it, polishes it, sends it to the Golgi apparatus. Golgi apparatus packages it up, sends it to where it needs to be. It needs to be sent in the cell. Okay. The mRNA's done. It's trash now. It's not going to get used anymore, so it gets taken out, torn up, eaten by the lysosome. Remember, my favorite guy. Okay. So I got some here, just a little bit of look at what we did. So if we had the dRNA strand, dRNA. So we have our DNA, TAC. We transcribe that into RNA, and we do the opposite. So TA, A used to be T, but now in mRNA there's no T, so we have U. C goes to G. The tRNA anticodon that goes with that is the opposite, UAC. And then we take that mRNA and we look at our chart to find the amino acid, and we find that it's methionine. And so you can practice these going through. And look at the PowerPoint and practice those going through. It's just a little bit of extra help. So this first step, transcription. All of this, translation. This going from mRNA to amino acid is translation.